Good afternoon for those in Europe and good morning for those in the United States, I guess. Welcome to what I think is the third or fourth webinar, the number 4 webinar of Prepare. Today's webinar has the title, Why Future Clinical Trials Will Be Platform Trials. This was one of the work packages in Prepare. And in 2014, we started with this plan of building a new type of study, a trial that would be prepared for the next pandemic. At that time, of course, we had no idea when that pandemic would start. And actually, we at that time also had not a very clear idea how such a study should look like. And I remember the words from Derek Angus, let's build a cool piece of science first. Well, we took some time for that, but I'm very pleased to say that that is exactly what has happened. I think these adaptive platform trials are really cool pieces of science. And the REMAP-CAP is an example of that, as is the principal study. And today I'm very pleased with the presenters of the webinars this afternoon, Derek Angus, Scott Berry, Chris Butler, and Lenny Derde. And they will address the basics of platform trials, of adaptive platform trials, a little bit about the statistics, the beauty, and then how these trials have functioned so far during this pandemic. So without taking any more seconds from your time, I'll hand over to Derek Angus for the first webinar presentation, Platform Trials, the Beauty of the Beast. Derek.
Perfect. All right. So yes, as Mark said, we were totally interested in being a little bit nerdy around trying to build some cool science. The motivation for this, which I think Mark didn't quite allude to, was this sense that coming out of H1N1, many parts of the scientific community had done a fantastic job of really coming together. The virus had been typed and fully sequenced and through cooperative sharing between virology labs around the world with dizzying speed. Papers were appearing in Nature in New England just within 2 or 3 weeks of recognition of H1N1. At the same time, even by the end of H1N1, we'd hardly managed to generate any evidence about how to actually take care of patients. In other words, there was a very tough time trying to do any, to generate evidence from randomized trials in the middle of pandemic. And of course, part of the reason for that is because although it's true all the time in medicine, it's particularly true in a pandemic that you're really trying to juggle 2 things simultaneously. While it's certainly true that we would like to find out what therapies help patients, we're also trying to treat patients as well as possible, and there's a sense of urgency and threat. And so these things almost get framed almost like a false choice. I think much of the world watched this amazing White House press conference about a year or so ago, where President Trump and Tony Fauci were asked about hydroxychloroquine. And one of the reporters said, so what should we do? Should we take hydroxychloroquine? And President Trump said, absolutely, take hydroxychloroquine. And Tony Fauci said, well, no, we'll need to do trials. And right there, there was this sense that, in a way, there was an imperative to try to do both things. You absolutely wanted to serve the patient in front of you as well as possible, and yet you wanted to generate knowledge as fast as possible so that the next patient after that would be better served by having learned from what you did with that first patient. This is, in fact, a classic dilemma of decision-making under certainty. It's known as the exploration-exploitation trade-off. And really what one wants to do is search for what is the optimal trade-off. That requires an integrated approach where you're trying to find the best trade-off or balance, where you're simultaneously trying to treat patients as well and learn as fast as possible. And in fact, outside medicine, organizations take responsibility for both parts of this ratio, for both the exploration, i.e. trying to generate new knowledge, and exploitation, i.e. exploiting all that you feel you know already. This is a cornerstone of decision-making under uncertainty. It's a feature of every complex adaptive system, and so it turns up in organization science, and mathematics, and evolutionary biology, and economics, everywhere. It's actually basically the cornerstone of reinforcement learning of artificial intelligence. It's how autonomous cars are learning how to drive. It is everywhere, everywhere, except in medicine. In medicine, we stopped taking single ownership for the ratio of trying to learn while doing and finding the balance, in part because we accidentally separated these things out. Now, it was well-intentioned. Things like the Belmont Report, the need to separate research from clinical care, felt like hugely important to discern the two differences. But the consequences were that we ended up building up separate organizations, separate cultures, separate people, funding, procedures, and goals. And that means that no single person or body or organization or institution is empowered to find the optimal trade-off. That's always true, but it became particularly obvious during a pandemic. So if we take, for example, as trialists, if we think about how do we generate causal inference, we still think that our best learning tool is the randomized clinical trial. But there are three major challenges in a pandemic. The first is that randomization itself is very uncomfortable for clinicians and for patients. The physician feels responsible for patient outcomes. The consequences are immediate and right in front of her or him. On the other hand, the clinical research project, well, it'll have a result sometime in the future, not quite sure how it helps my patient right now. The RCTs themselves are incredibly cumbersome. They can be slow to start, intrusive to execute. They don't produce answers fast enough. And then there's little coordination in the clinical research enterprise. Which trial should a hospital participate in? There are hundreds of RCTs registered for hydroxychloroquine. Few were ever completed. Academic medical centers have been bombarded with hundreds of requests to participate in trials, and there was no national or global prioritization. So what could the clinical research enterprise think about doing if it were trying to optimize the trade-off between learning and doing? That would involve thinking about clinical trial designs that lent into the realities of clinical care. For example, making randomization more comfortable, where fewer patients would be the guinea pig in the placebo or the control arm. So use a design with multiple arms, only one is control. Maybe use adaptive randomization, where you preferentially assign to a best therapy over time. Make entry into clinical trials one-stop shopping, where you simplify the interface between clinical practice and clinical research, using master protocols, and essentially trying to combine study questions into one learning platform. And along the way, ask ourselves how many of the cows of research are truly sacred cows. Placebo can add to scientific rigor, but the marginal gain from a placebo is not the same in every trial setting. In some settings, placebo may actually add relatively little to the rigor of interpreting the causal effect, but certainly adds a lot of burden. And so one poster child example of this would be trying to think about these so-called adaptive platform trials, of which we tried to design one, and we did design one under Prepare called REMAP-CAP. We're going to hear a lot about this in this morning's session, but I'm just trying to sort of set up why we have it. It's a global adaptive platform. It's designed with the idea that it could be running in the background between pandemics. So it's already running in the interpandemic period. And since the most common source of a pandemic is a community-acquired viral pneumonia, why not have the platform enrolling patients with severe pneumonia, testing multiple interventions simultaneously, and using something called response adaptive randomization under an overall Bayesian model, which we'll hear about from Scott in a minute. It would assess both the interpandemic and pandemic forms of pneumonia with basically preset rules to allow us to switch into pandemic mode. And that's exactly what we did. Now, the trial itself only really started getting going in about 18 months or so ago. So had COVID-19 been COVID-20, we would have probably been better prepared had we had a longer time to run in, but such is life. So just a couple of things, just again to set up, what do I mean by these adaptive platform trials? So in the world of RCTs, you can have something called master protocols, which is just you're trying to use the same form for common entry criteria, but you might be asking different questions over time. And then there's also something called adaptive trials that tend typically, for example, to use an underlying Bayesian design that would allow more flexibility in the kind of decisions that are made along the way in the trial. And then adaptive platform trials is really the cross of the two. They've typically been used in oncology. In the phase 2 space with relatively small sample sizes, it was a departure to think about using these as large-scale deployed, essentially comparative effectiveness trials in a pandemic setting. If we go back to I-SPY 2, because for 2 or 3 years, this was the first one we had seen published, in the middle of this design, a couple of papers were in the New England Journal of Medicine about 2016 or 2017. When you looked at the section where it described the randomization procedure, they had this little figure. Now, this does not look like a normal randomized trial. This is the way randomization was happening in the trial. It's called response adaptive randomization, and it's one of the key features of the inside of this trial that allows the adaptations to run, and that also helps the design to lean in to the questions being asked by clinicians. So let's unpack this for a second. Imagine if we were doing a normal trial, and at the start of the trial, you were unsure about whether treatment A or treatment B worked, and you were going to randomize people into treatment A or treatment B. And so presumably you started this trial because you were 50-50. Now, at the end of the trial, you end up saying, aha, A is better than B, because you're now more than 99% sure that A is greater than B. Well, let's think about what that means. Somewhere between the beginning and the end, you moved off 50-50. And you move towards nearly 100% sure. Now, that would mean looking under the hood. And we've all been raised in these typical frequentist designs where no one gets to do that. The DSMB does that. The trialists don't get to do that. Okay, so the trialists themselves don't need to watch it. But you could think about exploiting this information. Imagine if you had a planned trial of A versus B in 400 patients. And after 40 patients enrolled, you saw something like this. At this point, if it was in a normal design, the normal design is still taking the position that you're 50-50. No one's allowed to do anything differently. And yet at the same time, if the 41st patient was your uncle and you looked at this, if I were to pull you out in this virtual audience and ask you to raise your hands, are you all 50-50 about who would go into about which arm your uncle would get? I think every one of you would be saying, well, hold on a second. Am I in my uncle's will? How much did I like my uncle? And you'd be thinking about the fact that you had a strong sense that A and B were not behaving the same way. And you would be right. The probability that A is better than B is not 50-50. It's 78%. So imagine if you used that information, not you, the investigator, but if there was a preset model that as the trial went along, the model took these updated probabilities and then changed how randomization went going forward. So you're still rolling the dice, but imagine if you were weighting the dice so that now if you roll the dice in the next set of patients, if 78 out of 100 times it were to come up A, you would be pushing more patients into group A on the hunch that A may be better than B. And if that were true and you were right, you would get something like this. And after 80 patients, you'd be done and you could move to the next question. So there are some caveats. If the second 40 had been flat or going in the opposite direction, then the trial could just swing back to a more even 50-50 bet. If any of you have sat there and done trial design, you're sitting there and thinking, wait a minute, clinical sample size and power, it's always driven by the size of the smaller group. So this doesn't even make sense. And I would say you're right. In a 2-horse race, it's not clear that anything I've just showed you would matter. But if we had any question where we were trying to get treatment effects across different cohorts or with more than 2 arms, it starts to become very interesting because you don't want to necessarily spend all your sample size and power, asking every possible question with the same degree of specificity. You wouldn't mind moving towards the winner as fast as possible, and along the way, dropping the bad bets. So response adaptive randomization looks like this. You have a patient who starts out maybe getting 3 different choices, a third, a third, a third. As the trial goes on, data are accrued. These data go into a pre-specified model. And as that model starts to generate updated probabilities, not enough to stop the trial, but enough to start to take little bets. You could do things like start to push more patients into B and fewer into C. You can use it to trigger starting new arms or dropping arms. And you can even have different weights be generated by different pre-specified subgroups. So that's what we put together in a design where we thought we would have perpetual enrollment for continuous learning. It would be adaptive to match the odds of success to the odds of assignment. It would be multifactorial, where we would be testing multiple treatments simultaneously in different subgroups, yet trying to embed it both culturally in aligned with care decisions and in settings that use the electronic health record, leveraging EHR, and yet still having randomization for causal inference. And this is this REMAP design, of which REMAP CAP is the one we're going to hear about this morning. Just a little bit of nomenclature so that you understand the upcoming talks. Within REMAP, when you're asking any particular type of question, we call that a domain. For example, what's the right antibiotic? Or there could be a domain that is corticosteroids, where you're asking questions about steroids while simultaneously asking questions about antibiotics. And then within each domain, there's an intervention. So within the steroid domain, one of the interventions could be steroids at one particular dose. Another intervention could be no steroids. That would be one of the steroid arms. And then you can probably start to imagine, well, then wait a minute, patients are getting assigned to multiple interventions within multiple domains. And so we call that then a regimen or a recipe. And that can be different for different patients, and we can analyze within these different strata based on subgroups. And so then you see regimens, it's almost like choosing courses in a meal. Domain A could be the starter, and you could choose among 2 or 3 starters, including no starter at all. Domain B could be the main course. You choose among several main courses, including I'll skip the main course. And domain C is the dessert. And again, you could skip dessert. And you would be getting all your meals brought at the same time. So just to close by suggesting what would this actually look like in practice, imagine we were actually testing just 8 separate regimens. Now, REMAP CAP has effectively been testing tens of thousands of regimens. But even if we just had 8 regimens that were combining some antibiotics and steroids, and we were simulating in the real world some simulated cohort of what truth is. The trial doesn't know what truth is, but imagine if in truth, in an infinite population, 2 of the arms had a 20% mortality rate, much better than everything else, and the other 4 arms were much worse. On average, when we simulate these trials, you can see in the black solid line across the middle here that a traditional trial for the first 2,000 patients would put 250 patients into every arm. But this trial, these trials on average are putting into the gray bars, this is the sample size. They're loading all the patients into the 2 green arms. They didn't know the mortality rate, but as the trial was accruing over time, the trial was getting a hunch that these were the best performing arms. And so it was already loading patients into those arms, and it was protecting patients from the worst performing arms. Now here in red is essentially a proxy for power, and from the 2 trial design, you can see that there's not a huge difference in sample size, in the power from this sample size at this point. But we've got to this point in the trial with 80 fewer deaths. So REMAP designs are smarter in that they can consider many different treatment options and vary the options depending on the patient, and potentially safer in that they're probably playing what is probably the winner, such that if you're doing a trial in a setting where no one knows the answer outside the trial, on average, you're safer in the trial than out of it. So in conclusion, we're really trying to think about fusing clinical care with clinical research. This is to address the fact that this pandemic forces us to do 2 things simultaneously, to do and to learn. These activities are intertwined. We must learn while doing. Unfortunately, practice has traditionally been separated from research. The two enterprises, however, must lean into each other using learning designs that accommodate doing at the same time. And global adaptive platform trials have the potential as these LWD, or learning while doing, instruments. And I've gone over one example, REMAP-CAP, which you're now going to hear about in a lot more detail. So that's me. Thanks.
Thanks, Derek. Thank you very, very much for that. So just for the audience, we're going to hold the questions a little bit until all the presentations have been done. But please, if you have any questions to Derek, post them in the chat, and we'll try to go through them at the end of the presentations so that you get the answers that you need. And then it's my pleasure to introduce the next speaker. That's Scott Berry. So Scott Berry is the director of Berry Consultants, the stats team for REMAP-CAP as well. Scott, lovely that you're here. I'll hand the floor to you.
All right, thank you, Lenny, and thanks, Derek. So appreciate that, and this is really to tie in adaptive trials to webinars. I've been updating my slides as Derek has been speaking for this, and I've also updated the title since both Lenny and Derek were referring to this as the beast. I'll refer to this as taming the beast. So there's a lot of the word complexity has been thrown out there to this. So I'll try to give some description of what is the modeling behind the beast. It's a Bayesian modeling approach, and it leads to many things being done within the REMAP-CAP trial. So I'll try to tame the beast, if you will. And if move slide. So this is the beast. This is REMAP-CAP. The x-axis here is really time. As time went on, the line over here of COVID-19, everything to the left of that was REMAP-CAP that was built for community-acquired pneumonia. Let me move this here. We were enrolling community-acquired pneumonia in ICUs within the clinical trial, enrolling in steroid arms, antibiotic arms, macrolide, extending macrolide, and adding other domains. Each collection of those, each little bar is an intervention within a domain. We were enrolling patients in that, and we had built, before COVID-19, the pandemic was declared, we built what was called a sleeping strata, which was a pandemic strata within the protocol, that if there is a pandemic, immediately within the same sites, we start enrolling in the a group called pandemic. And we can start with the therapies we're already investigating, but we can add new therapies onto the trial to investigate them. And as time goes on, we can add other domains, as you can see happened in this trial. We are also stopping domains as information becomes available that certain interventions are good, bad, better than other interventions during the course of I don't even necessarily want to call it the experiment. This is, as Derek described, this is learning, confirming, and treating all at the same time as this learning process goes. That is REMAP-CAP. I'll describe there are adaptations within this that come from external information that can be adding to the REMAP-CAP trial. These are adaptations to the master protocol. So for example, as this trial, as the pandemic started, we were investigating steroids immediately in this. Then we added antivirals specific for COVID. We had antivirals added specific for influenza, but then we added immune modulation therapies, convalescent plasma, anticoagulation, heparin therapies. These get added externally into the trial and are part of the assignment to patients. And then there's information that's gained in the trial that triggers adaptations and triggers external information. And I'll talk more about those. But you can see during the process of this, in building this trial, we knew that this would happen, that we would add domains. And so we had to build the model knowing these were coming, even though we didn't know what they were. So we build a model that can take in all of these external domains coming in to investigate them. So this beast of what this is during the course of the trial, if a patient came in at any particular time, you can see the different domains that we were randomizing to in the trial. So early on in the trial, there may have been several domains that they were randomized to. And then midway through the trial, they could be randomized to 5, 6, 7 domains. So the same patient could be randomized to an immune modulation therapy, an antiviral therapy, anticoagulation therapy, convalescent plasma, vitamin C, statin, antiplatelet. I believe there were patients randomized to 7 different domains, the same patient to 7 different domains at different times. Then we started to learn which therapies were working, and you can see, for example, steroids within severe patients. We're not randomizing to them anymore. This is given as we learn that this is the optimal therapy to give. And in antiviral, it was to not give any antiviral. Now we're actually coming back and adding it potentially other antivirals. All of this was pre-specified that these adaptations could take place. And behind this, we need an algorithm to take this information, learn from it, draw conclusions, but also assign patients appropriately. So that's the taming of this beast. So the Bayesian, the Bayesian approach is what was pre-specified in this, and this is named after the Reverend Thomas Bayes, 18th century Reverend who posthumously actually published what is now, it was a theory of inverse probability. It's now called Bayes' theorem. For given information, how do I update my probabilities for each of the states of nature? This was critical given the adaptive design going on here. The frequentist, by the way, referred to as classical statistics, is much younger than Bayesian statistics, but that it's based on the design. Given my design, how likely is it that I would see this data? The problem in a pandemic is the design, we don't know what the design is. We don't know if we're going to get 10,000 patients, 500 patients, 100,000 patients. So the design is adaptive in and of itself. It's very difficult actually to do more classical statistics. So this is able to consume the data and make inferences about the state of nature. The adaptations don't complicate the statistical analyses of this. At the same time, we are dealing with potentially external data. We designed originally knowing that other trials are going to create information, and we don't want our trial that's making assignments to be naive to this other external information. So built from the get-go was the ability to insert external information into the trial, and the model needs to consume that for all of the adaptations that come out. All of these things led really to the only solution here, which is a Bayesian statistics approach, driving the adaptations, treating patients, consuming the complexity, and potentially consuming external information at the same time. So what are the internal adaptations to REMAP-CAP? And this is being driven by the Bayesian model, which, by the way, is the next slide. So I'll show you that. The internal the interventions, the individual interventions, we can declare them effective, which can happen when things are better than the nothing, the control arm within the domain. And that has happened in REMAP-CAP for tocilizumab, saracatinib, therapeutic anticoagulation in moderate patients, and a fixed dose steroids. That's in quotes, and I'll say a little bit more about the quotes. It can be determined to be superior in the domain, that it's better than anything else in the domain, not just the control, which would mean that would be the unique solution, and then that would be assigned within this treating process. That hasn't happened yet. If something can be found futile to the control within its domain, that's happened for convalescent plasma. It's happened for all of the antiviral arms, Kaletra, hydroxychloroquine, and the combination of the two, and therapeutic anticoagulation in severe patients. Those triggers were all hit from the data in the trial. You can be found inferior within a domain, so it's not just the control, but a different intervention is better than that intervention. So for example, anakinra could drop if tocilizumab is better than that, even though it might be better than the control, there may be a better one. That has not happened yet in the trial. Two things can be found equivalent in the trial. That has not happened. And of course, the adaptation could be keep going and use adaptive randomization for assignment. And that's happened many times throughout the trial as the adaptation is give me more information to the model. And this is the response adaptive randomization that Derek described. And in this circumstance, when an interim analysis is done, the Bayesian model creates what is the probability each combination of therapies, and as Derek described, at certain points in the trial, there was, I believe, 12,000 different therapies we could give to a patient. It calculates the probability each of those is the best thing to do for a patient and assigns them in proportion to that, and collects that data and then updates and continues that process. So behind this is the Bayesian model. So we built this Bayesian model to consume the primary endpoint, which is an ordinal endpoint of organ support-free days. This, by the way, in the CAP situation, it was 90-day mortality. When the pandemic was called, the sleeping strata was brought out. This was declared the endpoint for the COVID-19 pandemic. The model is an ordinal logistic model, which is typical for an ordinal endpoint, which also adjusts for covariates like age, sex, site nested within country, and a really important one in the trial is time. There are 2-week epochs during the course of the trial that we adjust. We expect that patients that come into the trial are going to have differential behavior through time. So the model's adjusting for that during the course of the trial. And then over here to the right are all the different interventions within domains and specified interactions. So in the model, there's an interaction between steroid and tocilizumab. So we are learning about the multifactorial aspect of many of these therapies. And down here to the bottom right is the endpoint within the immune modulation domain. And so the model's adjusting for this. The data goes in and the single model is being run for all internal adaptations, external reporting that goes on in the trial, and the response adaptive randomization. So for example, in the paper on the steroid result, and this was external information that triggered from the recovery trial that we stopped randomizing to steroid, but now we're able to report out the results. And in the Bayesian way, a 93% probability that steroids is better than no hydrocortisone therapy. In the tocilizumab IL-6 results that came out for tocilizumab and saracatinib, both of those with a probability that those therapies are better than no immune modulation, greater than 99%, greater than 99.9% and 99.5%. The Bayesian reading for those particular interventions. Meanwhile, the trial's still going, the model is still being run, Future results are coming out, by the way, are in preparation, and the readout from the model. Meanwhile, externally, we're adding new domains as we're interested in new therapies for the trial. So the unique scenario, let's hope it's unique. Let's hope we're not doing this yearly or every few years, but the trial is ready for it. Of the many therapies being tested in COVID-19, it was able to consume many therapies in a multifactorial aspect. The global urgency and the adaptations needed is as soon as we know the answer, we disclose that publicly, but we also implement that in the care of the patients that are coming into REMAP-CAP. The response to this and the ability to create conclusions, actionable conclusions in the trial, and behind this is the Bayesian modeling, and really the approach and the adaptations is really the heartbeat of the beast.
Thanks, Scott. Thanks very, very much. And I like your last remark that the Bayesian modeling is the heartbeat of the beast. The stats team has absolutely been at the core of the trial. So thanks. And again, for the people watching, we'll take questions at the end so that we can group them all and have a little bit of discussion amongst ourselves maybe. So for now, we'll move on to the next speaker. So Chris Butler. Chris is a GP in the UK, but he's also a professor of primary care at the University of Oxford, and he's the director of the clinical trials unit there, and he is the PI of both the ALICE trial in PREPARE and also the PRINCIPLE trial that are both the same type of trials that that REMAP-CAP is, so platform trials. So Chris, without further ado, I'd like to hand the floor to you.
Thank you very much, Lenny, and it's a great privilege for me to be on this platform, if I can use that word, with such eminent colleagues. And I'd also like to just pay a word of thanks to the leaders of the PREPARE consortium that really introduced me to the idea of platform trials. In particular, the coordinators, Herman Goosen and Menno de Jong. And it's really out of the PREPARE consortium with the far-sighted funding of the European Union that has led us onto the ability to do platform trials in the current pandemic. And we started out about, almost a decade ago doing a trial of oseltamivir for influenza-like illness, following on from the comment that has already been made that there were very few people randomized in the H1N1 pandemic. So we didn't really know if giving Tamiflu was a good thing or not. And we did a trial which had the capacity for adaptations as well as adding in new arms in 13 countries. And this was designed with the critical contribution of Ben Seville of Berry Consulting. And this is the kind of data that we were able to show that, on average, we found a one-day benefit. But given the trial design, we were able to show that there was a much greater benefit amongst older people who were sicker. And so with that experience of perhaps what might have been the first trial with the potential for response adaptation and adding in arms during the course of the trial itself, we set up the PRINCIPLE trial, a platform randomized trial of interventions against COVID-19 in the UK. And I guess important to mention that treatments in the community that keep people from becoming eligible to trials like REMAP-CAP are of critical importance. And, new interventions are coming down the line that will require evaluation, early treatment, and we can only do those trials in the community. So we sort of latched onto the idea of platform trial where we would have a standing master protocol and trial infrastructure that as they became available, we could add in new interventions. And as results became available, they could be announced and either for benefit or for futility or for safety, and could inform clinical care, hopefully during the pandemic itself. I think that was the main goal of PREPARE, that we would learn how to do trials, implement them, that would inform care within pandemics. So just to mention the PICO of the principal trial, we're looking for people basically at higher risk of a worse outcome from COVID, and presenting in primary care within 14 days of the onset of the symptoms. And we're comparing trial interventions against usual care without a study drug. And the co-primary outcome is time to self-reported recovery and 28-day hospitalization or death. And we have a range of secondary outcomes. And critically important here is the pragmatic nature of the trial. We've already heard about the embedded nature of REMAP-CAP, and what this means is we're trying to get this kind of study as close to usual care as possible without it interfering with the hurly-burly of delivering care under tough conditions. And you know, we're not asking the question, does a particular drug work better than a dummy version of that drug? We're asking whether, if you're a clinician seeing a patient in front of you, does it benefit that patient to add in a drug or treatment on top of whatever else you might be doing? So that's why we favor open design here. The platform trial allows you to add in and drop interventions as we go along. And Scott and Derek have already mentioned about response adaptive randomization, where more people within the trial can be allocated to the best performing treatments. So the proportions are changed. And in principle, the trial gets analyzed basically every week or every 2 weeks, depending on the recruitment rate. And this diagram shows how we have a flexible number of participants starting out to different treatments, interventions 1, 2, 3, 4, and 5. We give them equal numbers usually at the start of the trial. Perhaps at the first review, you can see more people have been allocated to intervention 3 because the solid section of the pie shows that more people are responding. And at review 2, you can see that the responders are, treatment successes are much greater now with treatment 3. So we can drop that and we can add in further interventions. And in designing the principal trial, working with just amazing colleagues, Ben Seville again and Nick Berry, we went through numerous simulations of what might the trial look like at different points of recruitment, and what might a meaningful benefit look like. And this is just one of numerous simulations that the trial team looked at to make some decisions about what might be a threshold for a probability of superiority or meaningful benefit. And here you can see the pink here is, in this case, a simulation around hydroxychloroquine, and the mauve is the standard of care. And we can see that the simulation at the first interim assumes 457 would be randomized to standard of care, 457 to hydroxychloroquine. And if that was the difference in terms of hospitalization estimates, then the probability of superiority would be 0.93, and the meaningful benefit effect, the probability of that would be 0.908. And then we can go on to the second interim and so forth. So the ability to simulate numerous different scenarios is critical to the design of the trial, and that early phase work can't be underestimated in its importance. Now, I've called this talk about taking research to where the patients are, and traditionally in primary care at any rate, we've waited for the patients to come to us, and if a participant comes to a practice that is open to the trial, then if somebody at that practice is aware of the trial, they can try and recruit them. But that denies people the opportunity who are not coming to that practice to participate. In England alone, there are 5,500 general practices. Not all of them are interested in wanting to become a trial site. And so if you limit participation by where you receive your health care, it's not what I call a democratic trial. So although we recruit very heavily through general practice and primary care, we've opened up this trial to anyone in the UK who is eligible. They can provide online consent. We can check eligibility online as well, and randomize them and courier them medication and study materials, and we follow them up either through telephone or online, that way making the study available to everyone who's eligible in the country. And we can recruit through self-referral, even through GP practices, through national helplines, and so forth. And we can also add to our follow-up of data through routinely collected data access. And this just shows you how we started out on the 2nd of April, just about a year ago, with hydroxychloroquine, as did many other studies. And through the good offices of the National Institute of Health Research Urgent Public Health Panel, this trial, in a sense, was protected. So we didn't fall into the trap of many other trials that were struggling to recruit, who were underpowered and who never gave an answer. This was a national urgent public, or it is a national urgent public health priority trial. And you can see we started recruiting into the azithromycin arm as the hydroxychloroquine arm was stopped by the regulator. And with the doxycycline, we've just, and we have readouts from those two, and we've just stopped recruiting now to budesonide, and next week we'll be providing, that's inhaled steroid, which is a kind of neat intervention, given that systemic steroids have been beneficial in hospital trials. We'll give a readout next week about the inhaled budesonide there. And recommendations from this trial have already flown into the NHS, impacting on care, and that speaks to the proximity of generating evidence to uptake in care that is possible from these findings, from these types of trials. And we've recruited now to be, in fact, I think the largest primary care trial of treatments for COVID in the community. And we started out because testing wasn't available, basing our recruitment on syndromic criteria. And now most of the people coming into the trial, the vast majority, do have a positive test for SARS-CoV-2, and that's reflected in the orange trial. We have recruited from general practices throughout the UK, even far north in Scotland and in Northern Ireland. And here are just some of the sample results with the probability of superiority here for doxycycline at very low figures, and you can see the hazard ratios not separating at all. So we can be pretty confident there's no benefit from that drug. And we can look at the subgroups as well to see if there's benefit in any particular subgroup too. So that just gives you a flavor of what I call now the UK's National Urgent Public Health Platform Trial with response adaptation. And, you know, just to pay tribute to everyone involved, particularly Ben Seville and Nick Berry, who've helped us so much with the design, and Michelle Detry with an execution team, and then everybody else who's helped with this study. So I'll leave it there, and thank you very much for your attention.
But I just want to say, I think it's amazing to kind of turn around the recruitment method and just ask patients if they want to be part of this trial, rather than kind of having just GP practices. Can you tell us something about both from the scientific community and from patients or patient panels, what kind of responses you get to that? Is that because I imagine it's well, it's innovative and it's also, I assume, very popular, but I was just wondering what the responses have been.
Yes, so people can get into this trial in many different ways, including self-referral. And we, in fact, are also able to approach people who have a positive test now. Okay, so that's a new innovation, and lots of governance issues have to have been negotiated for that, but that does allow us to call up selected people with a positive test in the right demographic and offer them the trial. And also clinicians in the general practices can search their databases for the incoming positive test results and then phone up patients and say, introduce them to the trial. And interestingly, almost universally, the patients are delighted to be approached and feel that they are being offered the chance to participate in research and be part of the solution as well and contribute their data for the benefit of those coming after them. So it's been a very positive experience. And, you know, in the past in Wales, the inverse care law was described by Tudor Hart, which goes that those most in need of healthcare are least likely to receive it. And in a way, we've turned the inverse research care law on its head, because, you know, those who are most likely to benefit from contributing to research, who have the most to offer, previously were the least likely to get the chance to be in research, and now we've turned that around. So it's a great innovation, and thank you for the question.
Thank you, Chris. I think we move on with the last presentation now, which is Marlene Dere, who you can already see. And then maybe you think, is she dressed up like a doctor just to do a webinar? No, she just came from the ICU where she's taking care of COVID patients today. Also maybe enrolling patients in REMAP-CAP, not sure today, but she will address the REMAP-CAP study, The Beast Unleashed. Lenny.
So the title of this presentation was REMAP-CAP, The Beast Unleashed. And you've already heard in the title of Derek's talk and Scott's presentation also the word beast. And that's because we had a lot of discussions in the last years about adaptive platform trials and how, you know, setting up one and running one internationally was really like taming a beast, exactly like Scott said in his presentation. And I actually used a similar title in a Prepare annual meeting in May 2019 that we had in Bologna. In that meeting, we also stressed the fact that, you know, it's very, very complicated. We ran into a lot of issues while setting up a REMAP-CAP, an adaptive platform trial. And at that point, the things that were most challenging for us were things like contracts. It was very hard to get contracts with sites, which is, I think, something that's recognizable for anyone who's ever done a clinical trial. The protocol was also challenging because it had to be modular. Because if you want the trial to be flexible and you want to add domains and interventions all the time, then you have to have a protocol that you don't have to completely rewrite every time that you do an adaptation. So that was something we struggled with. We struggled with how to structure the domains so that it was easy to implement new ones. Last but not least, we spent a lot of time on designing the CRF, and especially what we call the eligibility CRF. So that is more or less the randomization engine behind REMAP-CAP, where we ask a whole lot of questions. And in those questions, behind those questions, there's a lot of logic. And the logic makes sure that only patients who are eligible for a certain intervention or domain get assigned an intervention within such a domain. And that actually before the pandemic, at this meeting in Bologna, when we had 4 domains open, there were already about 8 pages of logic behind the randomization engine. So that's why that talk was called Taming the Beast. And at that time, we had a great couple of project managers that I called the superheroes, and they still are. And at that point, we had 3 for the European site, so under Prepare. And we had just started working with our colleagues from the UK, so Paul and Farah, and we also had the German team in setup. And then obviously we had the global project managers, Jane and Cam. And at that presentation, and remember again, this was May 2019, we said to each other that we were quite happy because we had what we called liftoff. We finally had, we were starting to get patients enrolled in the trial, which was, something that we were relieved about because setting up the trial came with a lot of challenges. It was not for nothing that we called it a beast that we had to tame. And at that point, we had 52 sites active across the globe, and we were very, very happy with that. And I'll now show you the last slide of that presentation, which was that, I hope that we would be a rocket in the next year. And a rocket meant that I was hoping that in the year between May 2019 and May 2020, we could include a total of 1,000 patients globally. So just slide is just to remind you, be careful what you wish for, because we all know what happened after that. The COVID pandemic happened. And that made us realize that, it wasn't such that we had to tame that beast, but the beast was unleashed. It had broken free from all its chains, and we really had to ramp up the work quickly to keep up with the demand. What I'm going to do in the next couple of slides is I'm going to give you an overview of a couple of things that we did since that meeting. And I'm going to refer back to the challenges we already had in May 2019. So contracts, everybody knows contracts with new clinical trial sites can be complicated. So in early 2020, when we realized that the pandemic was going to be, so that it was going to be a pandemic, that COVID-19 was going to spread outside of China, we activated the pandemic stratum. Scott's already talked about that. And we did that on the 3rd of March. So that's about week 10 of 2020. So in the 10 weeks after that, we were able to fix 116 contracts within 10 weeks. So that, I think, is an amazing achievement. And you can see that it luckily, the speed of that went down a little bit after that, but we are still creating new contracts with new sites every week. And we globally now have 310 sites participating in a trial, in contrast to the 52 about a year and a half ago, almost 2 years ago. The second thing is the domains. As both Derek and Scott alluded to, we work under a core or master protocol. We had a pandemic appendix to the core protocol ready before the pandemic started, and we've added a lot of domains. And for each of those domains, we've written a protocol document that we call a domain-specific appendix. And then some of these relate to what we call non-pandemic patients, so your regular CAP that only runs in ICU. And then we have the pandemic strata patients that are either moderately ill or severely ill, and without going into the detailed criteria, it more or less, not completely, overlaps with ward versus ICU patients. So as I said, we created a lot of new domains because we found that there were many, many questions to answer about COVID-19. And this gives you an overview of the number of new domains that we got ready in 2020. So this graph, this timeline goes from January 2020 to December 2020. And what you can see is that in that time period, we submitted and got approved 9 new domains. And what's not on this slide is that for most of these domains, there was actually a second and sometimes even a third version because new knowledge that came up required changes to the protocol, and that all these protocols were submitted as a substantial amendment across the globe. So for Europe, that is about 12 to 15 countries for each substantial amendment in a different language with different document requirements. So you can understand why I already called the project management team the heroes of this trial, but they've proven to be even bigger heroes during the last year. What we've also done is we've extended the group. So before the pandemic, we were active in Canada, the US, Europe, including the UK, and in Australia and New Zealand. But in the last year, we've added a couple of regions. So what you can see here is that we've added critical care Asia as a region, with sites open now in India, Nepal, and Pakistan. But you can also see 3 red dots, and actually 2 of these red dots have already become blue. So they've already become active in the REMAP-CAP family. So that's Colombia and Japan. And we are also negotiating with South Africa and Kenya to see if they can come on board the trial. And you can imagine that setting up the trial in these locations took quite a lot of work from quite a lot of people. And this is the recruitment graph of the last year. Again, all of this to show you that REMAP-CAP, it was a beast, it's still a beast, and it had to be tamed maybe, but it also got unleashed in the last year. So what you can see here in red is the graph for the kind of rocket style takeoff that I had hoped for in May 2019. But what you can also see is that very early in the pandemic, it became clear that we were going to match that prediction or go over that prediction. And we now have almost 7,000 patients included in the trial. And I say 7,000 patients, but because REMAP-CAP is multifactorial, and because, as Scott said, patients are randomized or can be randomized at least to multiple domains at the same time, this means that we have over 12,000, so almost 12,500 patient randomizations, of which the vast majority is COVID-19 patients, and that we are currently investigating or have completed 46 interventions in a staggering 14 domains. I think that is unleashed. And you can recruit patients and you can do contracts and you can build data management. That's all cool. But what about results? Did we get results? So that's why I'm showing you this graph. I think there's some data missing, but that's okay. So what you can see here is that when we recruited the first patients, so that was 6 days after we opened the pandemic strata, so on the 9th of March 2020, 6 months later, we had the first result. Scott has shown you the data from that JAMA paper that we published early September. And then recently, we published in the New England Journal of Medicine the results for tocilizumab and sarilumab, so the IL-6 receptor antagonists. Between November and November 2020 and January 2021, we also had several other platform conclusions. For instance, that therapeutic anticoagulation works in the wards, but not in the ICU, that convalescent plasma doesn't work in critically ill patients, and we also stopped including patients in the moderate state, mainly because of the recovery results, so external results, and we also had a conclusion about the therapeutic anticoagulation on the wards, that was effective. So here you can also see, very interestingly, that results for ward patients and ICU patients may differ. And then this one is gotten the wrong way around, but the antivirals, lopinavir, ritonavir, as well as hydroxychloroquine were proven not to be effective. And the red ones are still under submission. We hope to publish these results soon. The data for the therapeutic anticoagulation in the most critically ill patients has already been published as a preprint on medRxiv. So what else did we do? Did we only include patients and produce results in REMAP-CAP? No, I think we did a couple of other very important things, and that is collaborate. So I think what we knew is in the global pandemic, you have to collaborate, and we did that by collaborating with 2 other big platforms on anticoagulation, which means that everybody kind of had to put aside their ego for us to work together and draw conclusions from a combined, from an intervention, similar intervention on the 3 platforms. And I think that went very, very well. We also collaborate very nicely with the Lovett investigators on investigating vitamin C as a treatment in CAP and COVID. As I said, we've included new regions, which has brought a whole group of new great investigators to the trial. We've participated in meta-analysis, especially with the WHO. And we've also found a lot of people outside of the clinical trial community, but with expertise on certain interventions that we've really worked together with in a very good way, and that has helped the trial a lot. So all these collaborations and all this expansion and all this ramping up also means that we have to tell people what the trial is all about. So that's also one of the goals of this webinar, but to help people understand the trial and understand the results that we produce, we also have a couple of very short videos that you can view on Vimeo. And I like very much the fact that we didn't just put out the IL-6RA paper, but we did that while we did a free live stream hosted by Critical Care Reviews, where we explained the design of the trial, the stats of the trial, and then discussed the results with an editorial by Tim Walsh and a panel discussion. And I think that was really, really nice innovative way of sharing results. There are also a couple of podcasts that Critical Care Reviews Rob McSweeney did with several of the investigators. That's very interesting to listen to, I think. And that then brings me to the end of my presentation almost, where the one thing I want you to remember from these 15 minutes is that, this trial is a beast. It's an international adaptive platform trial, and the number of people working on it to deliver this trial and to deliver the things that we've delivered in the last 7 years, and especially in the last year and a half, the number of people is just amazing. And in the picture you see here, they are faceless, but they're not faceless to me. I know most of them, if not all of them, I'd say personally, but obviously we only see each other in digital meetings that occur much more often than they did a year and a half ago. And I really think we have to thank all of those people, all of the REMAP-CAP family, everybody who's included patients, who's helped us deliver this trial, who's helped us set it up. It's a huge group of people spread across the globe, and I like to call them my REMAP-CAP family. So a big thank you to all of you. I know many of you are listening, and if you want to follow them and the trial, you can see at the bottom of the slide where you should go on the different platforms. So with that, I think, is REMAP-CAP a beast unleashed? I think yes. We've proven we can build and continue a large global network. I think the approach of the adaptive platform trial has proven to work, not just REMAP-CAP, but also Principle, and also other trials that have delivered results in this pandemic are similar to or true adaptive platform trials. And I'm very grateful that the funding that we got through Prepare initially and now through Recover has made it possible for us to contribute to the treatment of COVID patients. And I hope we can continue to evaluate the best treatment for CAP for a long, long time. So with that, Mark, I give the floor back to you.
Thank you very much for all your presentations. I did enjoy them, so I hope the rest did as well. Before we go to questions, I do have a question, maybe for Derek and Scott, and that's about the primary outcome for the REMAP-CAP study, which is the organ support 3 days. We got a lot of questions about that. Could you a little bit about the pros and the cons of that endpoint, and if you had to do it all over again, would you still go for that primary outcome in your study?
Yeah.
Derek first, or
can you hear me? Am I?
Yeah.
So it behaves nicely statistically, and it captures something that is intuitively important, in that it is not only mortality, but also the burden of disease. And it captures it on a scale where mortality is worse than a lot of organ dysfunction, which is worse than a little organ dysfunction. And it gives a nice reward for someone who skates through COVID avoiding mechanical ventilation and respiratory failure. So those are all the strengths. You get readout sooner than you would in an instance where if you were enrolling low-risk patients who had a low chance of mortality but could certainly end up going on respiratory support, this single endpoint can give a readout in that patient population as well. So it scales across a wide range of diversity of illness for patients as well. So those are all the strengths. I think what's difficult about it is even though ventilator-free days has been in the ICU community for a long time, and even though things like infarct-free survival, these sort of combined endpoints have been very common in the cardiology world, people are still not that used to these so-called combined endpoints. So there's a degree of unfamiliarity. And then people aren't quite sure how to interpret what an odds ratio of 1.4 or 1.5 means. It's hard for them to sort of work out what that means. And then you end up trying to decompose it and trying to say, well, we saw this amount of effect on mortality and this amount of effect on organ dysfunction. But then in their minds, they're still trying to, they don't have a mental model for it. So the long and short of it is, it still feels like the right endpoint. It would have been nice if there had been a longer lead time to socialize everyone to it. I think if everyone had learned about things like this in medical school and been reading this endpoint for years, as don't think we'd be having this problem. I think it's largely a familiarity point, but I still feel good about what the endpoint is.
Yeah, I agree. Scott, do you wanna reflect on, you know, the interpretation and how difficult it is and what the odds ratio actually means?
Yeah, and I agree completely with Derek's description. The other thing that we're all a little operationally to receive that data and to get it right and to validate the data has been a bit of a challenge as well in the course of it. Though I do think it's, I'm glad we selected that endpoint. I think it's, had we done something a little less informative like mortality, I think it would have taken longer to get results. And I think we got the right results in this. So it is interesting. So people are familiar with an odds ratio when you talk about mortality or not mortality. What increasing the odds of that? What's interesting about this scale is when we talk about an odds ratio, it's across the scale. It's combined across mortality, avoiding organ support, avoiding longer organ support. So it could be referencing what's the chance of avoiding organ support for 10 or more days, organ support at all, or mortality. So it's an odds ratio combined across the scale. I think it's a very clinically relevant endpoint. It's just been harder, as Derek says, to socialize it and explain what that means to a clinician at the bedside. And I think some of that's on us to be able to explain it better and to be able to socialize it, because I think it has been a very clinically relevant endpoint.
I agree. And just for clarity, it's not just organ support for days, because if you die anywhere during follow-up, you are assigned a -1. So death is included as the worst outcome whenever you die. That's worse than any time on organ support, including 21 days on organ support. Yeah, I just wanted to say that because, you know, we're talking about this every single day, but not everybody may be talking about that. Scott, another question is, you know, you're doing a lot of interim analyses, and if you stop, you know, how do you know you're not stopping early for a positive result? And does it work the same way in this trial as in traditional RCTs? Can you reflect on that?
Yeah, so we are concerned before the trial started. In a traditional trial, we might know we're going to enroll 1,000 patients or 200 events, and we do interims at information fraction times, and we do alpha spending. People are used to this sort of thing. What's really hard in a pandemic is we don't know what the maximum size is. We don't know the design when we set it out. So what we did before the trial started is we simulated many different characteristics of the pandemic and created a rule that had very good behavior and was unlikely to have false positives. We simulated over the pandemic through thousands of patients our rule and our interim structure of a 99% posterior probability, and we knew up to about 2,000 patients within a domain that our type 1 error was actually less than the traditional 2.5% type 1 error at an intervention level. But we going into it, so we're concerned about not wanting to make a type 1 error, but the design isn't, this is the type 1 error and that creates your design. So we're very concerned about both types of errors. And so we simulate to understand the characteristics of this to limit the type 1 error, but it's something that we calculate and characterize in our design. It doesn't drive the analysis.
Very important. And I think we recently also started calling them adaptive analysis rather than interim analysis, to also kind of make clear that there's a difference, and that if we get to a platform conclusion because we hit a pre-specified trigger, then that means we are done, and we hit that pre-specified trigger.
Yeah, and we want, yeah, thank you for that. So we want to avoid the term interim because it's the on-time analysis. It is, this is the answer and this is the analysis, the adaptive analyses to this. As to some extent, we don't know what full sample size analyses look like. This is the on-time adaptive analyses. It's the result when we know it. And so the terminology is important. Interim is almost pejorative, as it's too early in the trial, and this is the on-time adaptive analyses when the conclusions are known in the trial.
Yeah, I mean, but part of the problem is trying to view a Bayesian design through a frequentist lens. It is useful without trying. You know, I've not quite had enough espresso this morning. It's at least the afternoon in Europe, but without trying to belabor the point from a sort of a layperson's term, frequentist statistics are basically saying, given that the theory is true, how likely are the data likely to be true? And so you're always suspicious of the data. Whereas Bayesian statistics are saying, given that the data are true, how likely is the theory to be true? In other words, Bayesian statistics has a theory, then observes the data, believes these data to be real, but nonetheless, they're only an observation, and then says, now, given what I've actually seen, how do I update my theory? But that's a flip of inference from the principle of a frequentist inference. And the problem is that we've been schooled in frequentist inference. And so therefore, the underlying principles that allow frequentist inference to be solid are different underlying principles. So in a way, we actually get worried about the wrong things. Like when we're looking at these Bayesian designs, we're worried about things that are threats to frequentist statistics. It's not to say that Bayesian is necessarily better, but we're actually worried about the wrong things when we're getting worried about, oh, the interim analyses. That's not where the threats lie in interpreting a Bayesian analysis. Would you agree with that, Scott? Or?
Yeah, no, absolutely. Absolutely.
So Mark, I don't know if you can see the questions in the chats.
No, I can't, but I do have a question.
Okay,
So,
go ahead.
also a question we have received. We are currently testing mostly repurposed drugs. Would the REMAP-CAP study be feasible to do investigational products for regulatory trials?
It would be, and it is. So, so it's a really great question. So in a perfect world, regulators would love the design that we have. But it has been true that when we've sought approval for registration grade components of REMAP-CAP, we have within those modules agreed to some parameters around the edges. So specifically, even though they're repurposed drugs, we are testing apremilast and eritarin. Eritarin is actually not repurposed. Eritarin is still only an investigational product, the anti-TLR4. Those drugs are in something called our immune modulation 2 domain, and it's called number 2 because that particular domain, if you're enrolled in that, including randomized to control arm, there are several things that happen. First of all, in order to appease regulatory concerns about the bias of open label, we are actually using placebo in that instance. So we're not putting placebo against everything. It's not heparin versus heparin placebo. But if you are randomized in that immunomodulation 2 domain, you get apremilast or eritarin or a placebo. And it could look like apremilast or it could look like eritarin. We don't give you double placebos. That would be like crazy. But there is no way to know if you're in the open label control arm. The other thing we did is we are going to report out basically a slightly different endpoint, which is being free of mortality or organ dysfunction by day 28. And then we've also said that we're putting a cap on the total size in order to help give a little bit more of a confidence around the so-called alpha spend, which is a negotiation around looking at it through a frequentist lens, some concerns about having too many looks. But it's all inside REMAP-CAP. And so it's all with the same inclusion and exclusion criteria. Oh, the other thing I should say is enhanced safety reporting for patients within that piece. So if you imagine it as one large learning platform, but if you're then hosting an investigational assessment, it's like that domain has all the normal parts of REMAP-CAP, plus we spike it with a little bit more safety reporting. We can spike it by having, for example, not having an open label control within that section. And we can spike it by doing things like that particular piece of the puzzle, we might turn off RAR, or we might cap the total size of that portion. So in other words, it is totally possible to host registration grade assessments for investigational drugs inside the REMAP-CAP platform. Not only is it possible, but we are actually doing it. It was one more beast, but it was possible.
Yeah, the multi-headed beast. It's interesting, Derek, because there's a question in the chat that actually relates to this. So it's actually a statement and a question, and I'd like you to reflect. So the person asking the question says, we need conventional trials to prove safety. So that's a statement. And then, but would you agree we also need conventional trials to prove a degree of efficacy before advancing them into platform trials? So in other words, are platform trials unsafer than other trials? And do we need to have some form of prior efficacy before we put interventions in a platform trial?
So I would actually start by challenging the statement. I have yet to see a conventional trial that has ever proven safety. Phase 3 trials are called efficacy and safety, but actually I don't know of any regulatory agency that thinks that safety is firmly established at the time that a drug is approved. That's why you have post-approval surveillance monitoring, and so safety is on a continuum. You're always managing the degree of safety. By definition, an event that occurs at, for example, 1 in 10,000 patients is unfindable in a phase 3 trial of just a 2,000 patient trial. So safety is always on a continuum. But I totally agree that you should always be looking at and evaluating safety. But actually, one of the origins of phase 2 trials was in the I-SPY 2 space, which was a phase 2 platform trial that could more efficiently spool through a number of candidates, so that you spent less time testing candidates that had no chance of progressing to phase 3. And they used interim endpoints. And it was essentially, what's the probability that if we took this drug forward into a phase 3, that it would be positive? I think if you remember that drug companies are taking between 10,000 and 100,000 potential molecules, and then finding out which one eventually gets registered. It's a huge winnowing process. And in that winnowing process, the idea of leveraging your bets is already built into the process. It's just that a lot of the time it's quite opaque. People make internal little business judgment calls about what moves from phase 1 to phase 2 to phase 3. Actually having a statistical scaffold that manages the relative probabilities of all the agents and gives estimates of which ones should best progress from one step to another could arguably be positioned as a more transparent and a more mathematically based evaluation process for all drug discovery, for the entire winnowing of what is both safe and most effective within a disease area.
Thanks, that's great, Derek. And maybe I can very briefly ask Scott to, just for the kind of non-experienced Bayesians, because you always say everybody has an inner Bayesian, to reflect on how we handle prior probabilities and what we can do so that we can incorporate information that is or is not already there before we start the trial.
Yeah, thanks. So the Bayesian approach is built on this notion of prior probabilities added to the data gives you the posterior probability, and that's what's being reported out in journals. So the answer that comes out of the Bayesian approach depends on the prior that goes in. In most of the cases of clinical trials, in many of the cases, the data swamps the prior and the prior doesn't matter much. But there are some circumstances where you want it to matter. So we have a built-in structure that if there's external empirical information, we can build that into the prior to be combined with the data in an explicit way written up front on how that's done. We did that in one circumstance of interferon combined with steroids. There was a concern about safety. We wanted that to be built in. Something very interesting, other parts is the structure to the prior is important. In some cases, you can say this therapy is independent of everything else. So for example, saralimumab and tocilizumab, they're both IL-6RA inhibitors. We expect them to potentially be similar. So we built a structure that if they are empirically similar, they would reinforce each other's effect. That was built in prospectively. So reading out the output at the end, that's built in. That same nesting or Bayesian hierarchical model was built into heparin in terms of severe and moderate. And within moderate, it could be differentially over subgroups of moderate. That was built in. So it can be bringing in external empirical information. It can be the structure of the prior is actually really important in the model itself. And so that was done in some cases. And then in other cases, like iridarin and apremilast, we specifically don't bring in external information, and it's all empirical in the outcome of the trial. And some cases we want prior information, some cases we don't, and in some cases we want structure. All that's possible in the structure of the Bayesian analysis.
Yeah, I think this notion of priors is important to talk about. So REMAP-CAP, just to reassure people, essentially takes the position that we don't have any kind of informative prior around a therapy already working better. That's, I think, what some people are sometimes asking. You could do a design, for example, imagine if we were testing interventions in teenagers, and teenagers hadn't been eligible until the drugs had been tested in adults. But we felt that teenagers might look like adults. You might say that when you launch the teenager trial, you use some of the information from the adult trial to begin with. So you have a hunch that the drug will probably work. And then as long as the effects are in roughly the same direction, you get to the answer quickly, which is advantageous because otherwise you might never get to the answer because the disease behaves differently, lower event rates and so forth. In REMAP-CAP, we're essentially neutral priors around treatment effects, but every trial makes prior assumptions in its structure, including Frequentis trials. When you sit there and you say, we're going to define superiority as a certain effect size, and we're going to put the following sample size on, you've essentially on day one ruled out the ability to say that a drug is superior when it has a treatment effect smaller than that. There's always implicit priors in the structure of every trial design, including traditional trial designs, and being more explicit about what those assumptions are, I actually think is an advantage in trial design, in this kind of trial design.
Thank you. I think we are already past half the hour, so let's do a last question. And the question was whether this platform trial can also be used for prophylactic therapy. Prophylaxis.
Yeah, so I think, so the answer is definitely yes, but a lot of it comes down to, I think in the first talk, I had talked about how some of the features really come into their own when there's uncertainty about whether whether a drug works similarly in different patient groups, and whether you might be wanting to test multiple different drugs at the same time. So I think if you're just doing a two-horse race of a Pfizer vaccine versus a placebo for a Pfizer vaccine, why don't you just do a 50,000 patient trial in 2 arms and do it as fast as possible? And the world will watch with amazement as these fantastic, very traditional trials from Moderna, Pfizer, AZ just raced to the finishing line. And it was a huge service for humanity. But if there were much more complicated prophylaxis issues, like, for example, passive immunization prophylaxis with monoclonal antibodies, where you don't know the dose and where you don't know whether it would be the same in immunocompromised patients or the same in patients who've already had a vaccine, but now you're wondering if they'd have breakthrough, you can quite quickly see there could be prophylaxis trials where there's uncertainty about which prophylaxis agent, whether to combine them, what dose to give, and whether you give the same dose to patients in different regions of the world exposed to different variants and so on. And as soon as you have that level of complexity, then a learning design that would quite quickly narrow the field and not keep trying to try agents that weren't working as well would likely come into its own.
Yeah, I'll just add to that the previous question about we need to do fixed or traditional trials leading into platform trials. Platform trials will be more efficient and faster to do any trial in multiple ways. For example, in the vaccine trials, imagine
once they get started, Scott.
What's that?
True, once they get started.
Yes, once they're started, the infrastructure's there, everything's faster, better data. Imagine the vaccine trials would have been Pfizer, Moderna, J&J, AstraZeneca, and placebo. We would have gotten readings for all of them faster. And in a pandemic where we care about 2 weeks and a month so incredibly much, that it would have been huge advantages. Now, I suspect getting that trial up and started would have been nearly impossible, and it was very quick to get the vaccine trials up. So yes, absolutely, prophylaxis could be done in platform trials.
Can I just say, well, I know we're over, but Scott and I just launched an adaptive platform trial in 21 days. And we did it in mild COVID patients because we used an endpoint that we'd already been evaluating in REMAP-CAP. You can move some of what was beast-like. Once you've learned how to wrangle the beast, what's been difficult about REMAP-CAP was everything was first, but we've done a ton of lesson learning. And I do not think, and I think it would be an important take-home point. You've made the point that it was a huge community doing a huge lift, and it totally was. But the benefits of that huge community doing that huge lift are in part that it won't be as daunting to do these platform trials in other areas. If you start in a brand new area with a brand new complicated endpoint, there's still a lot of work to be done. But in analogous areas, we can totally pivot far more quickly on the basis of the lessons learned, which again is a tribute to all the work. The work done by this REMAP-CAP family is not just serving REMAP-CAP, but it will also serve a faster timeline for launching other adaptive platform trials.
Derek, I think that's an excellent point to end the discussion with. Very excellent, because it kind of pulls together, you know, the beast that can not only be a big, you know, scary bear, but also a fuzzy, lovable animal that is now unleashed. And also, I think, you know, stressing the huge lift by many people and how we hope that that doesn't just benefit REMAP-CAP, but the community at large, and that we get better at doing this stuff, and that adaptive platform trials have a space in the clinical trials community. So thanks very much, both of you, for being here. Thanks also to Mark, who's not on camera anymore, and thanks to Chris, who unfortunately had to leave. Thanks to all the attendees for sticking around during this discussion, and thanks for all the questions in the chat box. Thanks, everyone.